Guten Morgen. I'm very pleased to be here to share my excitement about one of our spin-off company. We have a 170 spin-off company up and running. When I was asked to select one, I have no difficulties to choose because our company Conzeb is targeting the most critical issue of early detection of cancer. We have a lot of development in the cancer therapies, but if you don't detect early, then it fails. So, critical issue we are targeting. Second one, the technology they are using is ultra-fast, high-speed cameras, which I never heard about it before. So, important target, interesting technologies. Together with that, they have investment from somebody who did a $1 billion exit experience. With that, my last word is that seeing is believing. Let's welcome Kevin from Conzeb. Thank you very much.
Breaking the wall of biomarkers using extreme imaging. Kevin Sia, Conzeb.
Hi, good morning, everyone. So my name is Kevin from Conzeb, so representing the company to basically tell you our vision of how do we break the wall of biomarkers using these extreme imaging technologies. So the word I just mentioned here is actually referred to the common practice of the biomarker discovery, which could be used for disease diagnosis, treatment monitoring, or drug screening purposes. So why is it a wall, and what's wrong with that? So let me just basically do an exercise together here. So if I just tell you, and I ask you right now within seconds, can you tell me how many live dogs and dead bears? Can you tell that? It's quite difficult because not until you can actually see the entire image itself. But that's even more difficult because right now we are talking about to screen and look through every single images and many of them. And of course, you would ask, hey, I don't have to actually do an imaging here. All I need to do is to find a markers that could be representative enough to identify the dog here. Yes, that's right. You can see that right here. Every time you will see the dog, you will have a hand in front of that. So yes, based on this prior knowledge of this marker, you would have a chance to detect this dog, but not always. This example pretty much explains the two very important technical limitations of the biomarkers discovery in many applications. The first one is we don't have the imaging technologies that would be fast enough to screen and measure a large number of specimens, especially cells. Second, the use of the biomarkers are chemical specific, but most of the time it could be biased or even misleading, and not to mention the fact that they are very costly and very time-consuming to use. And what we are doing here is we try to use the extreme imaging technology, to address this problem. And this extreme imaging technology is 100 times faster than a scientific camera, and which is another 100 times faster than the best cell phone cameras that we are using. And because of that, that allows us to capture millions and millions of images of biological cells within 1 to 2 minutes. And combined with our microfluidic technologies, that we could actually image many cells in a single cell precisions and high resolutions. And besides that, we also do not use any chemical labels to image or analyze them. And apart from that, so instead we actually use deep learning methods to create a catalog of the intrinsic features that we extract from the single cells, and that could create an identity for each of the cells. And then you can actually study the properties and the functions of the cells, and that can be done in an automatically manner, an unbiased manner, and most importantly, is enormously large-scale manner, which is millions and millions of cells. So this actually really creates a great opportunity for us to develop a very versatile platform of cell assays, which is massive, intelligent, and label-free. So we have done several clinical tests and proof of concept experiments to check the utility of these label-free technologies. So, for example, this label-free method is sensitive enough to capture the circulating tumor cells a very small amount within millions of mouse blood cells. And also within millions of human blood cells, this label-free method is sensitive and specific enough to differentiate different immune cell types. And the same techniques, being label-free and intelligence, that could sensitively predict the cells response to the gene-targeted drug treatment. That's actually very exciting opportunities and excitement going on given by these technologies. And we are now having ongoing development and optimizations of the cell assay platforms and the deep learning analytics. So we are particularly focused on using these techniques to do what we call the imaging liquid biopsy for the cancer treatment and monitoring. And we are also working with the biomedical laboratories to use this tool to develop what we call the next generation single-cell analysis tool, so that you can have a better understanding of the single-cell biologies. So there's a lot of areas that we are working on here, and then we can provide instrumentations, assay kits platform, assays, surfaces. And very important is the enormous amount of single-cell image data that we can provide that can accelerate the development and research in biomedicines. And I hope that will convince you that extreme imaging technologies could benefit the biomarkers discovery to identify the live dog and dead bears in a very cost-effective manner. And thank you very much.